My pleasure to welcome Daniela Stock from the Victor Chang Cardiac Research Institute in Sydney, Australia, to speak today. And Daniela started studying mineralogy and crystallography at the University of Heidelberg and the Freie University of Berlin. And then she did a postdoc with Nobel Prize winner Robert Huber at the Max Planck Institute for Biochemie outside of Munich. And then she studied again with another Nobel Prize winner, John Walker, at the famous MRC Laboratory of Molecular Biology in Cambridge, UK. She then started her own lab in 2000, I think, at the MRC LMB. And in about 2006, she was recruited to the Victor Chang Institute in Australia. And I learned that very recently, when she's not working all the time, she has a new hobby where she is piloting an aircraft. So she knows a lot about motors. So.
Okay, is the micro on? Yes. Okay, thanks a lot, Mike, for the introduction, and thanks for coming. And I would also like to thank the APS for the invitation, and it's really great to be back here and to present our work of data that we have collected here a few years ago. So as you can see from the title, my lab is interested in molecular rotary motors, so biological rotary engines, and we work on rotary ATPases and the flagellar motor, and I'll tell you our most recent results about these projects. So to bring things into context, I like to compare these biological rotary motors to other biological devices and to other devices. So biological membranes are impenetrable to ions, so the only way ions can flow through a biological membrane is through gaps, so they are channels, and they have sophisticated gating and selectivity filters. So they are just like the floodgates in this hydroelectric power plant, so the ions or the water can only flow through if there's a gradient. So this is confusion controlled, and there is no energy involved. So if you would like to get the ions or the water back uphill, then you need energy, and in biology there are pumps. So they are A-linear pumps. They are either symporters or antiporters or P-type ATPases and ABC transporters. So they utilize either the energy of a counter ion gradient to provide the energy, or ATP hydrolysis to provide energy to pump ions against the gradient. So these are linear pumps, but they are also rotary pumps. They are the V-type ATPases, and I'm going to talk about them in a minute. And they are evolutionarily related to the power generators, the F-type ATP synthases. So they work very similar to the power generators in a hydroelectric power plant. I'm still looking for a better example of the flagellar motor that is a propeller to propel bacteria through viscous medium. Now, the best understood biological rotary motor is perhaps the ATP synthase. And this works remarkably similar to the power generators in a hydroelectric power plant. So here, the gravity pulls the water down the pipes, these pipes, and the pipes have turbines. Turbines have magnets mounted on top, and the rotating magnetic field induces electricity. So this is how man-made energy conversion works, and in biology, it works with the ATP synthase. So there's a proton gradient across membranes that's generated either by photosynthesis or respiration, and the flow of protons back through the membrane induces a rotation of the central rotor, and this provides the energy in the chemical reaction centers up here to make ATP adenosine triphosphate, and that, as most people here will know, is the universal energy carrier in biological systems from bacteria to humans, and a grown-up human turns over about his or her own body weight in ATP every day. I work at a cardiac research institute, so my line is usually our heart uses 6 kilograms of ATP every day just to keep the muscles pumping. But it's not just about muscle contraction, it's about cell division, protein synthesis, and so on. So every biological reaction basically needs energy input, and this comes in form of ATP hydrolysis. And so this is the main way to make ATP. So actually the chemical reaction center and how ATP is synthesized or hydrolyzed is quite well understood, and there was a Nobel Prize to John Walker for just the structure of the soluble part for that. What is still not well understood is how the turbine is driven by the flow of protons across the membrane. And this was the first glimpse of a biological turbine in 1999. And it showed that the turbine is made of subunits that consist of hydrophobic alpha helical coiled coils, and there's one very highly conserved residue, a carboxylate residue with a negative charge that sits right in the center of the membrane, and if this is mutated, then rotation stops. So this definitely takes up a proton and is involved in torque generation in the membrane. And so this is an electrostatic surface potential map, which shows in white hydrophobic residues, and blue is positively charged, and red is negatively charged residues. So the conserved glutamate sits right in the middle of the membrane. And so the charged protons can't slip through the center, so this is not an ion channel. And in fact, this central cavity is filled with hydrophobic phospholipids. And there's no other way a charged proton can reach this negatively charged residue than at the interface to a structurally unknown subunit. So this subunit here, called A subunit in F-type ATPases, is really the last unknown part of the whole respiratory chain, or the whole photosynthesis. And so of course we're working on it, but it is really nasty. So the currently most well-accepted model for the generation of rotation by protons assumes that there are two half channels at the interface of the ring, of the turbine ring, and the structurally unknown subunit. And it is assumed that the protons dissociate from the negative charge and expose it, and there is a highly conserved arginine residue sitting in the A subunit. And the idea is that these two opposing charges attract each other, and this is what mostly generates the rotation. On the other side, through the other half channel, due to the proton gradient, the protons come in and compete with the arginine, and so this goes on, and so this is a merry-go-round for protons. And there's a really nice animation from George Oster's lab in Berkeley that explains this a little bit more. So it adds some Brownian motion, and you can see that the protons dissociate from the carboxylate residue, expose the negative charge, then the negative charge gets attracted by the positively charged arginine residue, and here the incoming protons then compete, and so on. So in fact, all rotary ATPases contain two reversible motors. So there's the soluble part, which has an ATP-driven motor. So if you split the soluble part away from the membrane and add ATP, the central stalk will rotate, hydrolyze ATP. And on the other side, what I just explained, there are protons coming in. So if it's attached to the membrane, the protons will then run the turbine in the membrane, and this is upside down here. So then the torque generated in the membrane part will be then transmitted through the central stalk and induce conformational changes up here in the chemical reaction centers. And so these synthesize ATP from ADP and inorganic phosphate. Now to counteract rotation in these nucleotide binding subunits and in the A subunit, there are these subunits here called peripheral stalks. And so they counteract rotation. And whether these proteins run as ATP generators or as ATP hydrolyzing proton pumps depends on the magnitude of the transmembrane proton gradient and on the availability of ATP in the cytosol or in the matrix, and on the proton to ATP ratio, which is controlled in different organisms by different numbers of these of blades or subunits that make up the rotor ring in the membrane. So you can see here the conserved carboxylate residues are highlighted in yellow here, and different organisms have different numbers of proton acceptors in the membrane. So each one can translocate one proton through the membrane, so each one will provide energy. And so for example, in the bovine ATP synthase, you've got 8 blades in the turbine, so 8 protons get translocated through the membrane, and there are always 3 ATPs synthesized per 360-degree turn. So this gives 8 to 3 proton to ATP ratio. There's an organism called E. hyrae that has 10 of these proton acceptors, so that's a larger proton to ATP ratio. And plants, so spirulina, has 15 of these subunits. That's so far the record holder. So far 8 is the minimum, 15 is the maximum. And so I'd like to compare them to your gears on a bike. So in this case, let's say in the spirulina C15, that would be pedaling on your large chain. So you have to pedal a lot, but you can go uphill. Whereas here you would be on the smaller gear, high gears, so you have to pedal hard and you go faster. So this is one way organisms control the way or regulate the way their rotary ATPases rotate and at which ion gradient they start stalling and so on. So typically eukaryotes have 2 types of rotary ATPases, and one of them is the ATP synthase, which lives in mitochondria and is distinct or made to make ATP as ATP synthase, but there are also vacuolar ATPases that sit in most other internal membranes, and they are made to hydrolyze ATP and pump protons. So they are proton pumps that generate protons that are then utilized for other biological transport processes. Bacteria, however, mostly have only one rotary ATPase, and in most cases this is evolutionarily related to the F-type ATPase, but some few have V-type related enzyme that's also called A-type, because all sequenced archaea have these V-type related rotary ATPase. And so all the prokaryotic ones are supposed to be reversible and bidirectional rotary motors that can be adjusted, so they can either be ATP synthases or ATP-driven rotary proton pumps. Now, this might already be a little bit confusing with the F, V, and A-type ATPases, and the nomenclature of the subunits is really horrendous, unfortunately, but the one simple thing is that the hallmark of these ATPases is the peripheral stalk, and so the number of peripheral stalks is, determines whether they are F-type or A-type or B-type ATPases. So you just need to count the stalks, and then you know which you're dealing with. So for the past 12 years or so, we've been working on a bacterial V-type or A-type ATPase from the thermophile Thermus thermophilus. And we have purified this protein from Thermus thermophilus cells and tried to crystallize it, well, in the absence of any diffracting crystals of the intact protein complex, which is made of 9 different subunits and has a molecular weight of about 660 kilodaltons, and it has a rotary catalytic mechanism. So it's not easy to crystallize, but we managed in the first place to get a decent 23 angstrom negatively stained electron microscopy map from it. So this is how things looked like in 2004. And then we started playing this 3D puzzle by solving subunits at higher resolution, or other people did that. And so we docked subunits into the 3D EM density to build up a composite atomic model of the Thermus thermophilus A-type ATPase. So in 2004, 2 major things were missing. So one is, of course, the ion channel forming subunit that is still elusive, and the other thing that seemed to be much easier were the peripheral stalks made of 2 subunits called E and G. So they form a heterodimer, and there are 2 heterodimers of subunits E and G that form the peripheral stalks. And so now this is a crystallography project, but so it took many years. Unfortunately, it was meant to be an easy project to get a new research assistant started, and it took many years, and we went through several continents to solve the structures. So it crystallized relatively easily, but then the crystals had all kinds of nasty problems. First, they didn't diffract, and then we started mutating and going through, well, the crystallographers here will know what it means, go through different constructs, and then we didn't have enough methionines to solve the structure. with anomalous spacing. So we added methionines, then the crystals didn't diffract again, so we had to screen for more crystals. Then the next crystals that diffracted were twins, so it was a nasty story, but we eventually got a structure to 3.1 angstrom resolution using STAD on selenomethionine labeled protein on the mutant. And so to no surprise, we found an elongated structure about 160 angstrom long that forms a coiled coil at the end terminus of the two subunits, and then there's a globular head mostly formed by subunit E, which is shown in blue. And so for structural biologists, it was quite an exciting structure because it is the first example of a heterodimeric right-handed coiled coil. Some of you will know that coiled coils are very common in biology, but they are typically left-handed, and for example, the central stalk here is made of a left-handed coiled coil. So because we had the first right-handed heterodimeric coiled coil, we went ahead and did quite some analysis on that, and I'll go briefly through that. So left-handed coiled coils are defined in the protein sequence by heptide repeats. So every 7 residues, you've got a repeat of hydrophobic subunits that are aligned on the interfaces of the two helices and make the contact in between the two helices. Right-handed coiled coils can be defined either by a Hendekat, so an 11-mer repeat, or a Quindekat, 15-mer repeat. And the interesting things are the Hendekat repeats, because these 11-mer repeats lead to a very large pitch. So 1,000 angstrom, that means that the two helices are virtually parallel to each other. Whereas if you have a Quindekat repeat, it's the same pitch, but just opposite-handedness. So we analyzed the sequence of subunits E and G. So the yellow hydrophobic residues here make the interface of the coiled coil. And in subunit E, you can see that we have a Hendekat repeat throughout with a stutter here in the middle. Subunit G, however, starts with a Hendekat repeat at the end terminus, and so that leads to the two parallel helices at the end terminus, but then it switches to a Quindekat repeat, and that means that subunit G then wraps excuse me, much faster around subunit E. And at the very C terminus of subunit G, there's a kink, and subunit G then hugs subunit E. This will become important in a bit. So the next surprise with this project, which just kept being difficult, was that when we docked the structure into the electron microscopy density, on the side that we call the distal side, because it is away from the ion channel, it fitted really well. So you can see here that the structure follows the EM density. On the proximal side, however, it doesn't. So it sticks out at places and it just wouldn't fit. And you can see that here better in the animation, how it sticks out of the density. So the other problem was that we had a really nice data set, native data set of the unmutated structure to 2.25 angstrom resolution, but it should be simple to solve the structure by molecular replacement with a 3.1 angstrom structure in a different space group, but it simply wouldn't work. So there were some indications that there must be flexibility in the peripheral stalk structures. And so we anticipated that this was crystallized in a slightly different conformation. And my student then, Alistair Stewart, solved the structure the old-fashioned way by soaking these crystals in lots of different heavy atom derivatives, and he finally solved the structure then to higher resolution. And so here are the two structures, what we call PS1 to 3.1 angstrom resolution, and here's the new structure, PS2 to higher 2.25 angstrom resolution. And I hope you can already see from far that they are bent in slightly different ways. Now, the problems weren't over because the new structure fitted even less into the left proximal density. So then we lost patience and discovered an AMD and used the electron microscopy density as restraints to pull both structures, PS1 and PS2, into this density, and they kind of converge. So we call that our 23 angstrom structure. Now this is the 3.1 angstrom structure. This is PS2 at 2.25 and PS3 at 23 angstrom resolution. So that one fits into the proximal density. They all kind of sit on the same trajectory when you align them on their globular domains. And so there's some flexing involved and the twisting, and that leads to a maximum displacement of the two helices of 50 angstrom. So that's quite a lot. Then we discovered normal mode analysis. So I'm really an X-ray crystallographer. I've never used molecular dynamics before. And so we found this server called El Nemo on the web. We put our coordinates in, and so I'm sure most people here know better what normal mode analysis is than I do. You treat your atoms as masses and your bonds as springs, and then you apply some wave functions, and there's mode 1 to 6 are rigid body movements, and 7 and higher are wave functions or distortions. Excuse me. So normal mode 7 leads to flexing, and normal mode 8 to a twisting of the structure. So if we combine these normal modes, then we can actually simulate exactly, thank you very much, these three structures, and can interconvert them one from the other, and we get the same maximum displacement. So at the end, we really shouldn't have bothered to solve the second crystal structure. We could have just used normal mode analysis. And so you might ask yourself, what does it all mean? And what it means, I think, is that this really nice looking movie here is wrong, or at least slightly wrong, and we believe that the peripheral stalk is much more flexible than that. And to start explaining why I think this is, I need to go back again to 1999. I tried to make a very simple movie of catalysis in this so-called F1C10 complex of yeast ATP synthase that I had solved then. And because the soluble head sits at an angle to the membrane, if you simulate a rotation of the central stalk, the head just flops around and it looks really ugly. I don't think I ever showed this animation at the time. So Said Sanuga, who made this really nice looking movie, then straightened up the F1C10 complex and assumed that when the peripheral stalk that is bound in the intact protein complex serves to straighten up this complex. However, by now there are several of these F1C10 structures in the protein database. So there are 2 from yeast and 1 F1C8 structure from bovine, and none of them has the transmembrane axis aligned to the axis through the hexagonal head group. So they all have an axis to each other, so all the head groups are tilted towards the membranes. And so this is deemed a crystallization artifact, and no one thinks that means anything. However, if you look at class averages of electron micrographs, and there are no crystal forces involved that could bend the central stalk, then you can see that some of them show a tilt of the head towards the membrane. And I've drawn some lines through here, so they all look a little bit crooked. And if you look through all of these negative stained or also cryo-EM averages, you can see these tilts occur everywhere. So here and here. So I think they are true. So what does it all mean? So this is our composite model, just the, from the A-type ATPase from Dermis thermophilus. So we just put in the nucleotide binding domains and the central rotor, and this is the maximal tilt of the head that we get from the electron microscopy docking. Now, if we simulate a rotation while leaving the head group fixed, then we can see an eccentric movement of the transmembrane domain. Now, if we add the peripheral stalks and let them cycle through the movement that we obtained either from solving structures or from normal mode analysis, then they follow the transmembrane domain as if they can sniff where it is. So at first we thought, wow, this is surprising, but at the end I think it just means that these structures have co-evolved for billions of years, so the dynamics is really built into the sequence of these peripheral stalks. Now this of course isn't the full reality for several reasons. So one is that if you insert these ATP synthases into membranes, then the central stalk wouldn't make this eccentric movement, but it actually rotates around one axis. And that then leads to a wobble of the head group. And this is then again precisely followed by the dynamics of the peripheral stalks. Now, ever since I've seen this movie, which was done by Alistair Stewart, my student, I think, well, maybe I can retire now because I feel I understand ATP synthesis because it explains so much. So one, it explains the architecture of the peripheral stalks. So remember, at the very end termini, we had the 11 mer repeats with the 1000 angstrom pitch that leads to the parallel helices. And so just with my fingers here, that leads to flexibility in one direction, which means that the peripheral stalks can follow the wobble of the head, but they can still counteract rotation. So we call this the azimuthal rigidity, or, well, it's around the circumference. These two parallel helices will be rigid and not bend. However, if we move further up, so there's a Quinn-Deckard repeat that leads to a faster wrapping of these helices around each other, and that leads to a greater freedom of these helices that can now follow the wobble of the head group. And this is most pronounced here in this what we call the elbow region, where subunit G wraps around subunit E very quickly, and so that is then like a pivot or a cardanic pendulum that can follow the wobble of the head group. So I think we have finally found the reason for the architecture of the peripheral stalks. However, I think this movie explains more. So this concerted wobble, if you look at the interfaces of these subunits, so both of the E and G subunits and of the peripheral stalk subunits to the head group, then you can see that the contacts are mostly conserved. So there's no need to break any chemical bonds and reform them, as it is here in the central stalk. And, well, we think that at least contributes to the efficiency of these molecular machines, because breaking and reforming of chemical bond needs energies. But I think most importantly, if you look at it from far, I think it suggests that there is a possibility of crosstalk. So the subunits up here in the nucleotide binding domain will know what's going on in the subunits here at the entrance to the ion channel. And I think I've mentioned this subunit here already many times. So this is the highest resolution we have of the subunit A or I that forms part of the ion channel. And we think that, well, once the intact structure is solved in several different stages, then hopefully we can show that this takes part in this concerted wobble. So the bottom line is, the take-home message is that these ATP syntheses are much more dynamic and flexible than previously thought, and this could contribute to crosstalk. So I don't want to suggest that they work like pump jacks, but it's a little bit the same idea that they're joints that connect every part with every other part. Now, is this the same in all rotary ATPases? So the F-type ATPases have hardly any sequence conservation. There's no structure of bacterial F-type ATPase peripheral stalks there, but they are known to have a homodimeric right-handed coiled coil motif, and the eukaryotic F-type ATPase stalk is known, and it has kind of the same principles that down here you have two parallel helices, but up here the F6 subunit wraps around the pink subunit very fast and creates this elbow which will be flexible. And now these are electron microscopy data from John Rubinstein's lab and normal mode analysis. So we think if you compare the A-type ATPase on the left side to the F-type ATPase isolated from bovine mitochondria, on the right side, the features are the same, that the flexibility of the peripheral stalk is really very similar. Now, and so to our surprise, while we were writing up this paper about the dynamics of the peripheral stalk, we found this really nice animation from a group in Harvard on the web. So they had already implemented this in their movies. There's no publication about it. I have no idea how they came up with it, but they have already made the animation. Unfortunately, the rotation is in the wrong sense. It rotates the wrong way around, but it is really beautiful, and it also shows a feature from eukaryotic mitochondrial F-type ATPases. They are known to form dimers, but it goes even further. So there's some newer tomography data. So this is a eukaryotic mitochondrion showing the inner membrane highly folded into cristae, and this is where ATP synthases sit. And only recently, it turns out that the dimers of mitochondrial F-type ATPases actually form the ridges of these cristae and determine the angle. So you have these rows of dimers like batteries of dimeric F-type ATPase synthases framing the cristae. And this all comes from electron tomography data from Werner Kühlbrand's lab in Frankfurt in Germany. And so they have looked at different organisms. And astonishingly, so if you look at bovine mitochondria, the angle in between the dimers is different in different organisms. And so they came up with a really interesting idea, which is still not, it hasn't gone down well in the field, so no one seems to believe that. So they suggest that the electrical field is stronger, the stronger the angle is here. And that actually creates a stronger proton gradient, or that will drive the ATP synthase. And this, I think, is a good explanation why in bovine mitochondria you need only 8 proton translocators. So there's a smaller proton to ATP ratio needed to drive these ATP synthases in bovine than in S. cerevisiae, that's yeast, where you have a flatter angle. So they are, I think, very reminiscent to V-type motors. Of course, they are not piston engines, but rotary engines. But I think these angles here are very reminiscent to some fancy V motors in cars. So what does it all mean? So again, this comes down to the peripheral stalk. So the two dimers are connected via the peripheral stalks. And so I think there's a possibility then that this concerted wobble actually, that two dimers can crosstalk, can talk to each other, and that perhaps then also means that the dimers in the other direction can talk to each other. So this is not even a hypothesis, it's just my gut feeling that maybe all these rotations might be synchronized. What does it all mean? I don't know. But so these are the engines that provide the energy, and we also know that during aging, the shape of our mitochondria changes, so that might be due to the disassembly of these dimers in mitochondria, and so we all know we run out of energy when we age. So it might have some meaning, but I gave this talk in front of the bioenergetics community, and I think I go a little bit too far here, so let's see. So then I gave this talk recently in my institute, which is a cardiology research institute, so I wasn't expecting much feedback, but it turns out one of my colleagues is really into motorbikes. So he said, hey Daniela, your peripheral stalks are actually exactly like push rods. So we got together and wrote a really fun review and took this little machine apart like a macroscopic machine. So I think the central rotor is really like a crankshaft, and our peripheral stalks that might open and close valves at different angles and synchronize the whole machinery are like push rods, and there's the turbine and so on. So we'll see. But so I feel more and more that I really should have studied engineering rather than crystallography at university. Now, here's a collaboration with Carol Robinson. She is really the queen of mass spectrometry, and she can make everything fly to her mass spectrometers. And a while ago, she sent me a really excited email because they had tried for a while to get our intact 660 kilodalton A-type ATPase to fly through their mass spectrometer, and this is really difficult because this is a membrane protein, so it's packed in a detergent micelle. So it needed quite a lot of development, method development on their side. But so Carol is really excited about these spectra, and I'm not a mass spectrometrist, I don't know what it means, but they can deduce a lot of things. So not just the mass of the intact enzyme, but they can also do controlled decomposition, so you can get subcomplexes and answers of which subunit contacts the other subunits. But most excitingly, they're working now with two types of rotary ATPases, one from E. hyrae and the one from us, from Thermus thermophilus. And so they find different numbers and different kinds of lipids attached to the turbine subunits. And so again, I think there is some analogy to engines, because the lipids that you find are not the most abundant ones in the respective membrane of the organism, and they bind specifically to subunits, so they might help to smooth out gaps and probably prevent some proton leakages through the membrane. So the lipids are there as a sealant and as a lubricant for rotation. Okay, so now, yeah, the second part isn't quite that long, and it's about the bacterial flagella motor, which is another proton-powered biological engine, and this is all the brainchild of my former postdoc Lawrence Lee, who's now an independent group leader and following up his ideas. So, but it all comes back to ATP synthase in my life. So I already mentioned that bacterial ATP, F-type ATP synthases are reversible. So usually you have a proton gradient, the flow of protons drives a clockwise rotation, and that leads to ATP synthesis. However, in bacteria, if the ATP concentration in the cytosol is high, so there's a lot of fuel in the cytosol, and if the proton gradient drops, then they can switch into ATP-driven proton pumps. So ATP is hydrolyzed, and protons are then pumped from the cytosol outwards, and that means that the rotation changes to counterclockwise. So just to summarize this, ATP synthesis is coupled to proton flow towards the cytosol and clockwise rotation, and ATP hydrolysis to protons flowing out of the cytosol and counterclockwise. So maybe just a flick. So the flow of protons changes with the sense of rotation. So this is different in the flagella motor. So you will know that many bacteria have these hair-like filaments attached to their bodies, and this is another proton-driven rotary motor. So you can see the protons here indicated, and these animations were done by the Japanese television in collaboration with KG Number. And usually the sense of rotation of this motor is counterclockwise. So protons flow towards the cytosol, creating a counterclockwise movement that leads to an alignment of the filaments. And so the aligned filaments will then propel bacteria forwards. However, sometimes for 0.1 seconds, the rotation reverses to clockwise, and that leads to a disassembly of the flagella filaments onto a tumbling motion. So you can hardly see it, but sometimes they stop, tumble, and then shoot off in a different direction. And this is how bacteria steer through viscous media. So now just to summarize this, so here's the difference. Protons always flow in the same direction, but the sense of rotation can change. Okay, so here's an overview of bacterial chemotaxis. There's usually a signal that's received at a receptor, and that leads to a phosphorylation cascade, and that ends with phosphorylated KY. And phosphorylated KY then binds to the switch complex of the bacterial flagella motor, and that leads to a short switching to clockwise rotation and disassembly of the flagella filaments. And so this is how the bacterium changes its trajectory, so that the net motion is always towards nutrients and away from hazards. So there are about 20 or so proteins that build up the central machinery of the bacteriophagellar motor, and they all form rings. On here again is the switch complex, so here's phosphorylated KY. It binds to a protein in the switch complex called Fly-M. That leads to a conformational change in Fly-G, and that is supposed to lead to some changes of charges that are presented to the ion channel subunit, which also forms the stator. So this part would be over here. So my student in Cambridge cloned the 4 rotor subunits, Fly-F, Fly-G, Fly-M, Fly-N, and the first thing that crystallized was Fly-G, which is the torque-generating subunit. It sits just below the ion channel outlet here, just below the membrane. So it crystallized, and parts of the structure were already known. So the middle domain and C-terminal domain of this Fly-G protein from Thermatoga maritima was known, and we crystallized the full-length protein from Aquifax aeolicus. However, again, we couldn't solve the structure by molecular replacement, but this time it was relatively straightforward with selenomethionine and SAD. So here's the structure, and in addition to the middle and C-terminal domain, we have an N-terminal domain. So it is highly helical, 3 helical domains separated by 2 longer helices. And what was already known before, that the torque-generating helix is this one up here. It has a well-conserved separation, number of charged residues that are separated and lead to this charge separation, negative and positive. And it is thought that they are presented to the stator subunits. And somehow, in combination with the flow of protons, this leads to rotation. So the precise principles, how this works, are not known. Now, I already mentioned there are 2 structures now. Ours from Aquifax aeolicus in blue, and there's Thermatoga maritima in pinkish. And so luckily for us, a number of mutations are reported in the literature. So these red residues here are known to bias, if they are mutated to certain other residues, they are known to bias the flagella motor for clockwise rotation, so the bacterium tumbles all the time. And so, interestingly, there are 2 conformations in the C-terminal domain. So the endpoints of the pink conformation is from Thermatoga maritima structure, and the endpoint of the blue, rotated by 90 degrees, is our Aquifax aeolicus structure. And we think that the mutations actually prefer the clockwise structure, the Thermatoga maritima. So we think that the Thermatoga maritima is connected to clockwise rotation and ours counterclockwise. Now there are 2 sets of conformational differences and 2 sets of clusters of clockwise bias mutations. And so the second conformational difference is here in the middle domain. So the colored structure is our full-length Aquifax aeolicus structure, and this so-called open conformation is the Thermatoga maritima structure. And again, a cluster of mutants sits around this hydrophobic core, so they are all conserved hydrophobic residues. If you introduce any hydrophilic ones that would break this hydrophobic core, then that leads to clockwise bias. And there is an extreme clockwise bias introduced by deletion of these 3 residues here, so that would be like pulling on this helix here so that it would open. So again, we think that the Thermatoga maritima, the pink structure, is connected to the clockwise mode, and our structure, the closed one, is connected to the counterclockwise mode. So this is summarized here. So these are the 2 conformational changes. And the next story, unfortunately, is quite complicated, so I have shortened it. And so we have crystallized 1 monomer of Fly-G, which usually forms a ring of around 34-fold symmetry. And so the crystal contacts are mainly made by the so-called arm motifs of one subunit with the next subunit in the crystal lattice. And armadillo repeat motifs are very common in eukaryotes. They are common in signaling proteins like beta-catenin. And so there are hydrophobic residues that are conserved in beta-catenin, but also in Fly-G subunits. And they lead to a stacking of these trimeric motifs, and that leads to an extended right-handed helix of these arm motifs. So this is beta-catenin, but it is really the same in all Fly-G structures and crystal lattices. They are all of them are packed using these arm motifs. So we think it must have a physiological meaning. And Lawrence stared at this crystal packing for quite a while and assumed that this is the main contact in between subunits, then applied a curvature to have the right diameter of the ring. And so he came up with this model. So I don't know about you, so I thought this was terribly complicated and it looks nice, but I told him, look, we can never publish a model like that. We really need more evidence. And we knew that there was electron microscopy data around, so I said, Lawrence, why don't you just dock your model into the EM data? And so he did, and it fitted straight away, so without any further fiddling. So then I started listening to him, and then it got better because I found Dennis Thomas on a trip to Germany, and he kindly gave us his 3D EM reconstruction of a clockwise locked mutant of the Salmonella flagella motor, and Lawrence's clockwise model of the ring fitted in again straight away. But what is more compelling, I think, is even this side view here. So all the parts are in the right place. So here's the C-terminus that should sit where the exit of the ion channel is. There's the middle domain, which sits where it is known to sit from cysteine cross-linking studies, and there's the N-terminal domain, which is known to make contact with this protein here called Fly-F, which sits in the membrane. So everything sits in the right place. And so we then docked all other known structures, like there's a partial structure of Fly-M, we docked this in. It sits at about the right spacing for disulfide formation. And there's Fly-N, so we put a dimer of Fly-N in, and there are 3 to 4 Fly-N subunits in this ring-like structure down here. So fits all really well, but there's another piece of evidence. There's the EM projection map here is from a mutant that has the N-terminus of Fly-G deleted, and it is directly connected to Fly-F. So exactly the N-terminus is missing here, the density for that, and the Fly-F is pulled further down. So Lawrence's model really explains all the biology really well, but then also if you add these two pieces of information, the conformational changes and the ring, and apply the conformational changes within the ring, then we get this compelling reversal of charges on the torque-generating helix, which was initially just what we were looking for. So remember, the protons always fly in the same direction, but the flagella motor can reverse. So yeah, this is all. So now we are trying to prove it, and the open question, so the big conundrum is in the field is, why is there a symmetry mismatch between the transmembrane Fly-F ring, which has a 26-fold symmetry, and then the other switch proteins have a 34-fold symmetry, and here in the middle there's some disorder going on. So why is that? And it is not even known whether the location for the Fly-G is really here or maybe in this gap. So we need to prove a lot, and so we are trying to do again mass spectrometry with Carol Robinson, and hopefully electron microscopy and X-ray crystallography of larger complexes. So at the moment we're working on Fly-F and trying to get crystals of that and start hopefully building up the whole rotor complex. However, even if we manage to do this, so we try many different organisms, but there's a wonderful paper from Grant Jensen at Caltech. They do cryo-electron microscopy tomography with different strains of bacteria, and look how different they look. So perhaps there are different stoichiometries in different organisms, and so it all looks quite a bit different. And so this is it. I would like to thank Alistair's student, who's now a postdoc in my lab. So he did his PhD thesis mainly working on the peripheral stalk structures, and he was helped by Lawrence Lee, who was a postdoc, and his main project was the flagella motor, or the FlyG structure. He's now an independent group leader at our institute. And I would like to thank Carol Robinson's group for mass spectrometry. And last but not least, I would like to thank the synchrotron. So obviously, the APS, we collected a lot of data sets at GMCA and at BioCars, so 23ID and 14ID. And this was funded by the Australian synchrotron research program. And so now we've got our own synchrotron in Melbourne. So we are using that, and some of the very early data sets were collected while I was still in the UK, so at the ESRF. So thanks so much for your attention, and thanks again, Mike.